Hi, my name is Jack Nichols, and this presentation is on the role of telomeres in cancer. We'll start with a quick outline, which starts with a telomere overview, as well as an overview of telomerase, both of which we've learned about in class, but it's been a few chapters, so I thought I would review it. And then we will talk about how both of these things relate to cancer. So what are telomeres? Telomeres are sequences at the end of DNA that protect it from degradation. And you may be asking what kind of degradation. Well, these telomeres shorten each time they replicate because there are gaps left behind by primers that cannot be filled. And as we'll see on the next slide, which is an image, the 3' end of the chromosome overhangs the 5' end. And during each replication cycle, a small portion is cleaved off. And if the telomeres get too short, they will go into apoptosis. In this picture, we can see how the 3' end hangs over the 5' end, and we can see how these internal gaps, like in A, can get filled, but B and C cannot, and that's where telomeres come into play. If you wish to view this image longer or any of the other images, feel free to pause the presentation. Now, what is telomerase? Telomerase indicates cellular immortality because they're able to extend telomeres. They do this by adding repeats further down on the 5' strand, and then complementary base pairs fill in on the 3' strand. Now, at this point, the telomere has been extended, but there's a gap between the original chromosome and the telomere, and this gap is filled in the classic way with ligase and DNA polymerase. And this image is exactly what we just talked about on the last slide, and it can be found in our book if you would like to look at it more closely. Finally, telomerase is found in cells that divide repeatedly, and telomerase expression is either low or non-existent in somatic cells. So how does this relate to cancer? We would expect cancer cells to die quickly, relatively quickly, because of telomere shortening, because mutated cells are repeatedly dividing so fast, but that's actually not what happens. It's actually been found that in 85% of cancers, telomerase is highly expressed. This telomerase expression keeps the cell dividing and replicating nearly indefinitely. Because researchers now know this, they focus in on how to manipulate telomeres to gain some sort of control over cancer cells. They've isolated telomere homolog oligonucleotides as a potential cancer therapy. These T oligos are specific sequences that mimic that 3' overhang, like we saw in one of those first pictures. And since they're very short, they signal to the cell that the telomere has become so short that it's ready to go into apoptosis. Essentially, what's happening is that it's tricking the cell into thinking that the telomeres have become so short that the cell is ready to die. Interestingly enough, this process is mediated by p53, which is a checkpoint protein that we learned about in class. T11 is a specific T oligonucleotide that researchers have been working with. We can see on the bottom left the actual base pair sequence of T11. And not only is T11 good at essentially tricking the cell into going in apoptosis, but it also has a number of other benefits, making it a promising cancer therapy. For example, it upregulates tumor differentiation, which inhibits angiogenesis. It increases the effectiveness of radiation therapy. And for reasons that researchers have yet to elucidate, T11, for some reason, only affects mutated cells. It doesn't touch healthy cells at all. And researchers' best conjecture is that the physiology of mutated cells have changed in some way that T11 can differentiate between. So what's next for this therapy? Because high telomerase activity is present in such a wide range of cancers, T11 seems to be a really promising approach. Current research is focusing on its exact mechanism of action, how it affects cancer cells specifically, and ways to stabilize its delivery into the nucleus, and so many factors can affect its composition. Thank you for listening to this presentation. My citations can be found on the next slide. And if you have any other questions, please feel free to either reach out or comment on the discussion board. Thanks.